Hello everybody, you're going to experience the most boringest thing so yeah, I'm gonna be explaining the central dogma of biology, aka transcription and translation, by answering 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14 questions. Yeah, question 1. Explanation. That's not a question. But yeah, explanation of DNA structure. DNA is a linked strand, and which resemble a twisted ladder, aka a double helix. Each strand has alternating sugar. Yeah, this right here. You see that? Yeah, you see that. Each strand has alternating sugar 3 phosphate groups, each attached to each, and also attached to each group is one of 4 bases, aka the A, T, C, G. Over here, there's a molecule. I'll show you where it's right there. That's a nucleotide. That's what makes up DNA, RNA, and that stuff. How does RNA polymerase know where to start and stop copying? Well, it knows how to stop and start copying signals in DNA called promoters. I'm gonna look that up so I know. Promoters. What is that? Called promoters. It tells RNA polymerase where to begin and stop replicating. Where does transcription take place in the cell? It takes place in the nucleus. Right. This thing right here. Whee! What is a primer? A primer is a nucleic acid sequence that serves as a starting point for DNA synthesis. DNA polymerase enzymes are only capable of adding nucleotides to the 3' end of an existing nucleic acid, requiring a primer to be bound to the template before DNA polymerase can begin with a complementary strand. And apparently has a temperature of 52 to 58 degrees. I'm pretty sure that's like how it... What is RNA polymerase? It is RNA polymerase. It creates mRNA out of the template strand. Wow, look at that. I understand that perfectly, and I think you should too. mRNA. What is the structure of mRNA? It is a long single-strand molecule made up of nucleotides, with the AUGCs, or the bases, and it looks like this, and it's used to make proteins. Well, what happens between mRNA, between transcription and translation? A little thing called RNA splicing, which what happens is a little bit of RNA splicing, which involves the removal of certain sequences referred to as intervening sequences, or introns, and exons are what's left of that splicing, and they form the final part of mRNA. Translation occurs. It occurs in ribosomes. What is the structure of these ribosomes? Well, glad you asked. It looks like this. Wow, and this too. That's so cool. What is the structure of tRNA? Well, tRNA is a distinctive folded structure with 3 Harpin loops, don't ask me what that is, that form a shape of a 3-leafed clover so that the codons and anticodons connect. The folded structure is to do also happens because of hydrogen connecting to some complementary bases. So it looks like this. It looks something like this. The anticodons go here. I'm going to explain that soon. This is like when you hear people say that it has an L shape, this is what they mean. This is probably like the simplified version. The amino acid will go like around here, and that's about it. It will go into a ribosome to read mRNA to make amino acids. Yeah. What are codons and anticodons? You see that anticodon thing. They're codons. You need to know that first, I think. Codons come in sets of 3 and make up mRNA. So if we go back to mRNA, we go here, images, here. These 3, that would be a codon, I'm pretty sure. And anticodons are a set of 3 opposing codons that help make amino acids, and I think also make up TRNA. Yeah. Oh, that's what it means by I need to do more research. Yeah, it's made up of these loops. The that's what the anticodons are. Oh my gosh, I'm learning as I'm recording. What is that? Anyways, what are amino acids? Amino acid. Yeah, amino acids are molecules that combine to form proteins. And there are 20 different types of amino acids. Yeah. And how does TRNA make amino acids and enter the ribosome? Except in a solvent accessible channel at the subunit of the intersurface. So I'm using that little tube, I guess. Yeah. It enters the ribosomes, lines up with a matching mRNA, and spits out the correct amino acid using those codons and anticodons. But what happens to this created amino acid chain, which looks like the amino acid chain folds up into a protein. That's its entire purpose. And what are the 4 protein structures? There is primary, secondary, tertiary, and quaternary. Wow. Look at that. Oh no. Pretty epic. And that's actually it. That's the central dogma of biology. See ya. Thank you for watching.